Right. I think, should we give a few more minutes or shall we start with the introductions now?
1 minute and then you can start.
Okay.
Because a lot of people are just getting in.
Mm-hmm. Okay, so good afternoon, good morning, good evening, everyone, and welcome to the EMN Webinar of the month. It's my pleasure to introduce today's speaker, Dr. Fidele Tugizimana. He's a biochemist and internationally recognized leader in metabolomics, working at the interface of systems biology, artificial intelligence, and translational science. Dr. Tugizima has started his career with a degree in philosophy from Urbaniana University in Rome, alongside a master's and a PhD degree in biochemistry from the University of Johannesburg, a very multidisciplinary foundation that generally shapes his scientific thinking. And he currently works as a leader in research science at University of Johannesburg in South Africa, where he's also a director of the Research Center for Plant Metabolomics. He serves as a specialist science at Omnia Group, and he's the current president of the Metabolomics South Africa and a board member of the Metabolomics Society. And most recently, and fresh from the oven, he has just been selected for the Metabolomics Society President's Award. So congratulations, Fidele. He leads a very diverse research group and has mentored over 30 postgraduate students and postdoctoral fellows through his career, driving cutting-edge research in computational metabolomics, AI, and multiomics integration, with applications in sustainable agriculture, natural products, and health innovation. Please, everyone, welcome Dr. Fidele Tugizimana. Sorry if I'm not pronouncing it correctly. Yeah. And just a quick reminders for the other speakers to stay connected with us through the EMN through the social media platforms. Can we go to the next slide, please? Yeah, so please follow us on LinkedIn, Twitter, and reach out through our email if you want to connect with us. And also a quick reminder for everyone to register for the webinar we're having next month with Dr. Monica Kala. So Fidele and Monica are both going to be at the MedSoc conference in Argentina, and we all look forward to seeing you all over there. And now, without more delay, welcome Fidele, and we're looking forward to your presentation. Thank you for being with us today.
Thank you so much. Let me share my screen. Can you see my screen?
Yes.
Presentation model. Thank you so much for the invitation, the EMN, and thank you for those who are in audience listening to the webinar. And it's good always to come back home. I was in the EMN some years back. It's very, very nice for the activities that you organize in the course of the year, and to really be the voice of the early career and give the opportunities and the platform where the early career we can interact. So for today, the presentation for this webinar, it's beyond the spray. It's to talk about the biostimulant and also how we use metabolomics, or we can use metabolomics to understand how this formulation works. So before, just to say where I am located, the lab in which I work, which is situated at the University of Johannesburg, in the Department of Biochemistry. Really a very dynamic group, and most of the work, it's really the group that does the work. So we have three main research areas, biostimulant and plant interaction, natural product for drug discovery, and also methodology or methods used in metabolomics. For today, I will be focusing on this part of the work that is done under this research area, and mostly of this work is done with a Omnia group, where I'm also affiliated as my second home. Also, the work we do in our lab, one of the things that drive the research questions or the solutions that we try to formulate, it's really to have a science that has impact on African problem, African challenge, African context. And there is this African Union framework, that aspirations that have been formulated that guides decisions or activities or initiatives that different layers of society can have to try to improve lives in Africa. However, in what we do often, most of them, it's really in global collaborations, be the collaborators that we work with, but also some of the questions or problems that are of global scale that we would want to contribute to. Before talking about biostimulant, I would like to just, it's not really, I'm sure most of you are aware of basic definition of what is metabolomics, but I like to always start by this background to say, okay, when we speak of metabolomics, what are we talking about? I would like to understand metabolomics in terms of the biological system and looking at the two spaces in a biological system, a gene space and the chemical space. And the metabolomics really deals with the chemical space, but doing so, measuring the small molecules known as metabolites, we can gain quite a lot of information on what is happening in the gene space, but the same also what is happening in the environment outside the biological system. Apologies. Then when we look at the metabolites at this multi-layer of different biological information, where the metabolites are on this level, we can understand metabolites not really as just end product, but they are really contribute to different regulation events that happen at other biological information layers. So metabolomics gives us really this snapshot view of what is happening at the chemical space, but at the same time, be able to understand functionally what are the molecular events that are happening in any biological system under investigation. For today, we talk about biostimulant as the topic of the presentation is stated. We all know agriculture is a must in our survival as a human. So we really rely a lot on agriculture for food security, but also it does affect different aspect of human life economically and also in the environment. However, we are aware of different changes that are happening on a global scale in different parts of the world. And this is an old slide, but if we were to think of today's, the current situations, agriculture faces quite a lot of challenge. In addition to population growth, but also how can we produce sustainably enough food that can feed all the population. So the FAO had indicated that how a drastic increase in food insecurity for in the last 4 years, from 2019 to 2022. This, I'm sure, for the current situation could even be worse. And the question then comes, what are the solutions? So one of the solutions is how can we formulate or have a program that is lead to the sustainable or precise precision agriculture. One of them is the use of what is known as biostimulant. Okay, the current technology that can help, the data analytics that can help, all of these to help for crop management, resource utilization, and the better decision making. However, when it comes to biostimulant, I'll come back to this. How this formulation works, it is still very enigmatic. There's quite a lot of questions around it, and that affects the formulation, the incorporation of biostimulant into agriculture practices. So I did ask AI about the biostimulant market, just something designed from using AI. Despite those questions around the mode of action of biostimulant, we really can see that the biostimulant market is really growing exponentially. So there is really interest in incorporating this formulation in agriculture practices, and there have been some benefits that have been observed, and it is projected that this growth will really reach closer to 36 billion US dollars. So that's quite a lot. And in different regions of the globe, the market is really growing also in a different exponential. However, that still leaves those questions that how can we improve the formulation of this biostimulant? How can we innovatively create a new formulation? And how do we use this formulation in agriculture? So what is biostimulant? We can understand biostimulant as any substance that helps the plant to grow, and also without necessarily being a chemical or a nutrients input. So some of the understanding so far is that the biostimulant do stimulate the different biological events in the plant, and which leads to improved nutrients absorption, enhanced stress resistance, and other physiological events that leads to growth, promotion, and also hopefully great yield production. However, to be able to implement or incorporate this in agriculture practices, we really need to understand how this formulation works. And that understanding also can help the biostimulant industry in a way they can develop new formulations that may lead to better results. I did put these slides here just to bring up also another concept that has been in the biostimulant space, what they call biostimulant 2.0, where the intent is to mix different formulations. In this case, for instance, where you can have microbial biostimulant, either mixed together or microbial biostimulant mixed with a non-microbial biostimulant. And the question remains, how will these two type of biostimulant, how would they work together? What are the results? What are the mode of actions? So this is still a question mark, but there have been attempts that have been done, and there are a number of studies that are being done to try to understand. Also in our lab, we have done a number of studies around these lines. So what we do in our lab, we use metabolomics, of course, to try understand or decode some of the element that can lead to an understanding of mode of action of this formulation. We do investigate both microbial biostimulant and non-microbial biostimulant. In this case, would be humic substances based or seaweed. And for microbial biostimulant, what we have been looking at, investigating bacterial-based PGPR and fungi. So we apply metabolomics, and I just borrowed these slides from Renata, where she designed different components that involved in the metabolomics workflow, and also adding the emerging tools that are being developed to improve the annotation of metabolites, which is one of the bottleneck in the metabolomics workflow. And that helps us really to decode what are the events that can really lead to understanding of how this biostimulant works. We do, of course, add some physiological or phenotypic measurements that helps to understand what is happening on the molecular level is translated to observable benefits on the plant. If we look then in the literature, this is also a bit old, 2021. Within those 6 years, we already saw an exponential increase in publication using metabolomics to study biostimulant. So there is really interest in using the omics sciences that can help us to decode this mystery, which I can call in this case the enigma of how this formulation works and how do they promote growth. And with that, then how can we make better biostimulants? So there is an article that was published a few years ago where they coined the phrase that, yes, metabolites possibly holds the key in a way we can understand how, in a way that this formulation works. So for today, I did say computational metabolomics, but I would like to situate this also in this artificial intelligence age where we are. We see indeed quite a lot of interest that these AI tools that helps in a way that we do science, in our case, metabolomics or any other omics. And it is really at different levels that we apply these machine learning, deep learning tools that can help us to do our research and get to better insight. So in an unprecedented way. So we, working in a biostimulant sphere, we did also propose some methodological steps or methodological output that can help in a way anyone want to study the plant. This is just a method that was published in one of the environmental metabolomics book, where we describe the use of metabolomics to understand how the plant respond to a stress. In this case was a controlled drought stress, and also how you can use computational metabolomics to understand signaling events or regulatory events that are involved in biostimulant-mediated growth promotion or abiotic stress tolerance in plant. So these are method chapters do provide some steps that can be applied to any crop and in any environment. So for today, I will use two studies to illustrate the use of metabolomics to understand or to study biostimulant. So the first study, which is mostly focusing on microbial biostimulant, and where we really looked at the microbes based formulation. And the other one, it will be a field study that also was designed to understand the mode of action of a biostimulant. So microbial formulations often are applied on the roots, or they are sprayed on the leaves, but mostly they are applied either on the seed, and then different ways of applying those formulation, and we see event or the benefits. So microbes, we know there is already a root of a plant. In the rhizosphere, we have already a number of microbes that lives there. And within the plant also, the roots, in endosphere, we have also a number of microbes that lives there. These beneficial microbes do interact with the roots and the plant, and they activate a number of events that lead to the promotion, to a growth promotion, enhanced resistance or defense mechanisms, or even priming event. Priming understood as conditioning the plant to respond better in subsequent stress conditions. So one would then postulate that this communication between microbes, roots, the plant, because the plant also do release some metabolites known as exudates, and also the endophytes. So there is really a complex or a network of chemical communication that really leads to the events that we see. So here then we pose the question, okay, if possibly these interactions are driven by this chemical communication or chemical intercommunication, what are the chemistry of these microbes that could give us a clue in terms of this communication? So this study then focused on mapping the chemical profile of Bacillus strains. The way we did, just in a simple in vitro, so it's just a liquid culture, and we profile the intracellular metabolome and the extracellular metabolome of these Bacillus strains. So these strains, it was a combination, a consortia of 5 strains, and also individual strains that make up those that consortia. The study was done by Renata and in also collaboration with Carl and Justin from Netherlands and the UK. So culture and measured the growth curve that helps us to select some time points that we can measure that snapshot of the metabolome. Samples were analyzed on the LCMS. We use LCMS mostly, and then applying this molecular networking and other tools to be able to try to annotate that chemical space of the strains. So what we observed, it's really a number of different superclasses, different metabolites that characterize both intracellular and extracellular metabolome of the bacteria. And what was interest for us was this class of lipopeptides that we really enhanced mostly towards the late stage. So these lipopeptides, this class of compounds, have different functions in the rhizosphere and also on the plant. In the rhizosphere, they do sometimes play a role of recruiting other microbes, so the biofilm formation, and also activate certain events in the plant. So one, in that language, thinking of the chemical language that is involved in that interactions, in that communication, this could be playing a key role in those events. So the co-culturing of those, putting together those 5 strains of Bacillus in that one consortium, we saw that it does indeed promote the synthesis of those lipopeptides. So the single, I didn't show the figure here, but the single strains, the profile of those lipopeptide in the single strains, could see that in terms of level, relative quantification, they were less compared to the consortium. So the consortium in that case could be something that would be better to use on the plant. So this really a side project from that microbial metabolomics, where we did contribute to this microbe mass tools development, which is really used to help in annotation. You are able to really annotate microbial metabolites and be able to tell which type of microbes that produce a specific metabolite. But this was done with led by Peter Dorsen lab. So that microbial biostimulant, I labeled here just PGPR, that consortium of 5 bacillus strains that we did profile. We did a number of studies on that formulation in a greenhouse. So then we could postulate, based on those different studies that we did, we could postulate a certain hypothetical model that can explain how that consortia, that formulation works, the mode of action of it. So we did measure the metabolites in the leaves, in the tissue, and also in the sap, which is really a vehicle through which different nutrients or metabolites that are exchanged. And also we did some gene expression and methylation. So we saw that really all that integration of different measurements and absolute quantification, because this was a targeted analysis that we did based on the untargeted analysis, where we got some key pathway and key metabolite of interest. Then we had to validate, confirm using targeted analysis, targeting certain classes that you had observed here. So we saw that the application of this consortium, HPR here refer to that consortium of 5 bacillus strains, does really affect a number of key pathways, both in secondary metabolism and also in primary metabolism, and also which correlates also to the physiological events or measurement that we had made that correlate to enhanced antioxidant defense, lignin biosynthesis, energy production, controlled respiration, and increased in shoot root biomass. So there is very positive effect. It could correlate to these different events, mode of actions of this consortium. And as I said in the previous slides, so some of those key chemical clues or lexicon that is involved in the interaction, one of them could be those lipopeptides that we could see that way in high levels. However, it's a model generated from a controlled environment greenhouse studies. And the question becomes, okay, under field condition, what happened? Is it the same scenario? Do we see the same? Is the model can be validated under field conditions to understand that mode of action? So we did also a field trial. So it's the same bacterial, the same microbial biostimulant formulation based on the 5 bacillus strains. So we did a field trial, and then we harvested at different time points, and one of them be the transition stage or vegetative reproductive stage, then at the reproductive stage. And also we did measure the yield towards the end and the protein content to understand, also to assess the nutrition value related to the application of biostimulant. So the biostimulant that was used is that 5 strains consortium. So we use again the LCMS as an analytical platform and the annotation. There's a friend of mine who says if there is no PCA, there is no metabolomics, but anyway. So, but in these PCA score plots, we could see that indeed there is effect of the treatment. If we see the treatment here, the two time point that we see, we can see the cluster, the sample groupings that are observed, that are happening. So which point to a differential or different metabolic profile in these samples. So the annotation, these are some of the classes that we annotated in terms of the metabolites. Then we then go deeper to try to make sense of what we observed. So I will just share with you this example in this case, where we see a number of change in the oxylipin, and which we can postulate that interaction of the microbe and the plant roots, and that leads to activation of defense-related mechanisms, but in a sense of defense priming. So we looked into other different pathways that were impacted to make sense, and this correlated really with what we saw in a greenhouse. So then we postulated the model that explains the mode of action of that biostimulant. Also, this just looking at the leaf metabolome. We did correct also roots that we could study what's happening at the roots metabolome level. So there are different really changes in the metabolome of the plant that we could see that relates also to different physiological events. And with the agronomic trait that we had measured, we could see that is improved root growth, also the beneficial nematode, so the improved soil health because of that biostimulant that was applied, and also improved leaf biomass, and also increased yield. We saw increased yield and the protein content of the crop. So these molecular events that we could see indeed were translated into phenotypic traits that are beneficial to them, that we can really measure to say this biostimulant indeed has benefit to the plant in terms of growth promotion, defense priming, because of these molecular events that it activates as a mode of action, and it can be seen. So that microbial biostimulant formulation, it's now a product that is on the market called Backstim 100. Is of course by Omnia, and I just said business is increasing or the productivity is increasing, the farmer is happy, and the Omnia also then can explain how the product is working. And based on that insight, a company like this that is involved in biostimulant formulation, it can guide them in a way they can formulate or develop other formulation. That's the case of this study that we did, where also now we looked at different consortium now of the bacillus, different formulation, different combination. And we also see this was also in a field-grown maize. We saw some benefits, but the three consortia, they work in a different way. So they activate different events in the plant, and then the study still needs then such information needs to be then taken further to design better dose formulation, or if they need to be taken further to the market. So with this, really just as illustration how we can use metabolomics in biostimulant industry formulation, what type of information that Metabolomics provides. And as you saw, when possible, we do integrate other omics layers or targeted gene expression to be able to really get a full picture that explains as accurately as possible how this formulation or activates cellular events or molecular events that can explain what we see phenotypically. So these are the two studies I wanted to share, but as I said, we do work also on non-microbial. There are two studies that one study we concluded on the seaweed, and there are other studies that are going on seaweed and the humic substances, also looking at the mode of application. Should we the spray versus a drench or root application, what are the differences and how the plant perceives those applications. What we also do is either to look at the formulation itself. In this case, as I've shown, where we looked at the backstem, which is made of 5 bacillus strains. We looked at it from a perspective to understand the chemical space of it that can give us an idea in terms of different chemical language or chemical clues that are used in interaction. And also to look at the plant, how the plant responds, that also can provide information in terms of the mode of action of the formulation. So I being helped by AI to put some of the key panels that can really define how metabolomics can be used to understand or to decode the mode of action of this formulation. Of course, there's experiments that is done, and at the end, what we want really, it's a model that can explain the changes we see, or if no change also, to go back to the drawing board in terms of product formulation or product optimization. Then we do a number of validation and translation that can really get a product, for instance, like backstem on the market. And as I started saying, for a sustainable and increased yield production, because at the end of the day, in agriculture, we want to really address the food security, but also the impact on the environment and also on the economy. So this, very briefly, what I thought to share with you, and there are conversation, questions, input. I'm really open. I would like to end with already a gratitude to the team that does much of the work, a lot actually, and also the collaborators, but also the people who assist financially so that we can do this research. And thank you to all of you who are listening. And if you have questions, I'm really open. Thank you a lot.
Thank you very much for this great presentation. We do have some questions here in our text, so I'll read the first one. What do you think is the greatest challenge for implementing biostimulants in the agricultural practice of South Africa and or in other parts of the world? Is there any examples of case where you have worked from experimental trials all the way to implementation in the agriculture industry?
Yeah. Should I answer, or do I wait for more questions? Or
No, you can answer that.
Okay.
We can go to the next.
So there are a number of challenges. They are still there. Firstly, I think apart from Europe, in Europe where they did actually tried to formulate a phrase, a kind of legal-oriented framework, regulation-oriented framework of the biostimulant that they can be put on the market. So far, the biostimulant are still regulated under fertilizers regulations. Reason being, that what I started saying, the mode of action, the clarity, what are these products, what are these formulation, and how can you ensure the quality of that product that you put on the market, etc. So there are still those questions of understanding what is biostimulant, what is in the formulation, what is good, what is bad, and so that challenge is still there, and so far they have sold under the fertilizer regulation framework. So the study that I showed, where we looked at that by microbial formulation from what it is in terms of just of what the chemistry that it contains, it has, and also the greenhouse trials that we did, then also the field trial, and then the product has been put on the market and it's being used here in South Africa and also in Europe and in America. So some of the challenge then, the first one still the understanding of the mode of action of the biostimulant, and which then have implication in terms of regulation, in terms of incorporation. And the other challenges, I think, is the industry itself, the biostimulant industry. How do we develop a product? What would be the best product to formulate? Some industry, of course, is agriculture, but also there is a business component. You invest a lot into in developing a product, but what is the return on investment? Should we take that long route of metabolomics that takes years, etc., or what is the shortest route that can be done? So those are still questions, but we see interest every day when I open Google Scholar. I see at least 5 papers that are talking about biostimulant. So there is really a growing interest, and I believe some of those challenges will be addressed as the science and the industry grow.
Great. Second question is, are there known metabolite markers associated with successful endophytic lifestyle establishment bacillus inside plant tissues, leaves, or roots in the maize plant?
Again. Say again, Renata, the question.
Are there no metabolite markers associated with successful endophytic lifestyle establishment bacillus inside plant tissues, leaves, or roots in the maize plant?
Okay, so we haven't looked into endophytes yet, and I have a colleague who have been encouraging me to look into it, into these endophytes. It is something good that I would be interested to look into, but I guess at some point we get that to design a project to really investigate into the endophytes, which because I believe the microbiome of the plant, it does play quite a big role in the health of the plant. So far, what I presented or what we have been working on, just the microbe outside, but to look at that interaction and also that establishment of the endophytes in the root tissues or other parts of the plant. We haven't looked at it, but I believe there would be some markers that can help to be able to track or to confirm that successful establishment or the benefits of how it works in the plant and the benefits that it brings to the plant. Yeah.
Okay. Next is, what class of oxylipins were reported in the Nephali study? Did you do any further? What class of oxylipins were reported in the Nephali study? Did you do any further validation of these molecules using target methods?
Yes, we did. No, we haven't done any targeted analysis for those specific oxalipine. It is something that I hope we can design a project to look into, because we saw indeed a number of classes of oxalipine that were here they are listed, that were as differentiating markers, if I may put that way. So it was non-targeted analysis. It would be nice really to look into this. Non-targeted analysis will generate quite a lot of hypotheses, yes, and some hypotheses led to formulating those hypothetical models, but it leaves us with some hypotheses that we need to investigate more to understand indeed what is happening, like the previous study way in a greenhouse that was also untargeted, non-targeted analysis. Then we got some classes of compounds. We looked into them with another study with targeted analysis and added gene expression, methylation, to really understand at the molecular level, to the whole layer, what is happening. Yes, we will be really looking to this oxalipine and see indeed what's going on.
Okay. Can you comment more about the use of seaweed? What are the main species used for this practice, and where are they harvested from? Seaweeds.
Okay, so the seaweed that have been worked on, I did work on. Some of the things that I report here, unfortunately, I'm not allowed to disclose most of it. They are business company related. Yes, the one of the Omnia, it's known as a kelp, which is on the market, but the kelp is also it's a solution, a formulation that is on the market from a company called Afrikelp, that is based here in South Africa. And they have also other seaweed formulation that we are working on that unfortunately I cannot disclose because it's the product they are working on. And yeah, but maybe when we finish the project, they agree that okay, we'll publish with those specific information. Yeah.
Okay. Do you think future microbial biostimulant development will shift from strain-based selection towards metabolite-guided design?
I believe so, because like this study, no, yeah, that one, where we have different strains combination in this consortium 1, 2, 3, and we see different divergence in terms of functions. So, and also in this study, we tried to see key markers that differentiate the 3 consortium. So that can be something that can guide the formulation to say, we have these key metabolites, markers that drives the 2 differences that we see in terms of different formulation, but also the way they intercommunicate and the benefits that the product we want to formulate, if it is for growth promotion, if it is for stress response, or which specific stress, and also would it respond, here it was maize, would be the same case for all crop. Yeah, so this, I believe, yes, I agree, these metabolites markers that we can be able to annotate and guide, they would be helpful in terms of product formulation.
Okay. Which machine learning approaches are currently most effective for linking metabolite profiles to biostimulant performance?
Yes, that's one of the questions that we have had when we finished this project was we measured things separately. So we did metabolomics on its own. We would do if any gene expression on its own, and also this agronomic or phenotypic trait measured on their own. Then we simply, by observation, do a postulation. It would be nice in the same way we want to integrate different omics to integrate the metabolomics results with phenotypic measurements or physiological measurement. That's something that also we are looking into. Will there be a method that can be used to be more accurate in terms of you correlate what you measure? I don't have it here. The figure where we tried to look at do a simple correlation, metabolite and the physiological measurements that we would have done, but it would be nice if, what algorithm or method that can be used to generate such correlation. And I foresee as we generate this data, data increasing, we may really be able now to generate predictive models that can link to different profile, to different traits, and that would be very great. Yeah.
Next one is awesome work. Thank you for the insightful presentation. It's fundamental research for shifting agriculture into more sustainable practice. Is there a lot of pressure from the traditional industry of chemical fertilizers to suppress this kind of research?
At the first glance, one would think so. But I believe actually with the shift in terms of the way the world, the global trends, where we are pushing sustainability, I guess the industry also is shifting. Of course, biostimulant will never replace those, how they call them, NPK, those fertilizers, nitrogen, phosphorus, those first elements that the plants need. As I said from the start, by definition, biostimulants are not fertilizers, they are not nutrients. They stimulate the plant or help the plant for nutrients accumulation, nutrients use efficiency, etc. So they won't replace the really chemical fertilizers per se, but they may balance in the way we use those fertilizers, or also the way we use other chemical formulation to assist the plant, pesticides, fungicide, etc. If you have a plant that is primed to defend itself against a number of biotic stresses, you may not need to use pesticide, for instance. So it's a shift not to replace completely those NPK formulations, but to complement and to really move towards sustainable practices. Yes, we may reduce some chemical formulations that are used in agriculture, but not really those essential nutrients that the plant needs. Yeah.
In microbial biostimulants, which metabolite classes are currently considered indicators of successful plant microbe communication under abiotic stress?
Which chemical class?
Indicators of successful plant microbe communication under abiotic stress.
Okay, so I would be careful to generalize because this model that I've been showing, it's built on maize, where we used maize. So, and remember, this communication between the microbes and the plant, it's not one way, it's two ways. The plant itself does secrete in the rhizosphere, for instance, exudates a number of metabolites that affect that microbial community that is there, and not the plants are different, right? And the microbes, the soil where the plant is would be different. So I would be cautious to really generalize that the specific plant and metabolites or class of metabolites, apart from the traditional, the classical hormones that we know indoors, acetic acid and other growth promoting hormones that we know in the as modes of communication between the microbes, the bacteria, the PGPR and the plant, but the other class of compounds, it can be very microbe dependent and also plant dependent in that, and the soil chemistry in that triangle communication. Yeah.
Okay, we have one last question here. So thank you for the fantastic presentation. Professor, I'm incredibly intrigued and impressed by how seamless your lab handles complex metabolomics data analysis. As a student diving into this field, I'm currently struggling with GNPS molecular networking. While the raw processing tools are great, I find it incredibly challenging to handle data clutter and get the final network layout to look proper and biologically interpretable. Do you have any practical advice, workflow suggestion, or specific plugins you recommend to students for cleaning up and optimizing GNPS network visualization?
Okay, that's an interesting question. Yes. Okay. So for the GNPS, let's say one of the tools in the GNPS will be molecular networking, the classical feature-based. The idea or the philosophy behind is to improve the annotation based on understanding the fragmentation that would have done and the similarity, and yes, and then assuming you can really put together different spectra that have similar fragmentation pattern, and that helps for annotation such in the library. But the molecular networking has a limitation and advantage. Advantage being that we're now in a position where we can really know beyond the known. What I mean is, if I'm able to know one metabolites in a cluster, I can speculate or try now have an easy way to look further for the unknown nodes or spectra to be able to decode the structure, the identity of the metabolites. So it does have the advantage. Disadvantage, it's also spectra library dependent. Depending on the library that you have searched, there is the probability to know A, B, C, D. So they sometimes may feel that you click and you get an answer, it's upload and get the answer, but it still requires you, the scientist, to sit, understand the basics, the principles of how the data that you have acquired, what were the parameters, the cleanness of the data, the accuracy, and your mass measurement, all those aspects related to the data acquisition, because those are the ones that also that will help you to select correct parameters when you upload your files in your GNPS, so that you can get a possibly good output. Then the visualization is mostly done in GNPS2. You can still do the visualization there, but often the tendency is just to do visualization with the cytoscape. That's really a skill, that how to use the cytoscape for the visualization. It's really it comes with the training by doing it, and the more you do it, the better you really understand what to do. So that's just annotation. Then to move to the biology, then once you have assigned a name to the signals that you have measured, at least it's a step towards a biological interpretation. So there are different tools that you can use, be it pathway analysis or network analysis. Those also, it's another exercise. If you say, the person said the student starting, yes, be patient with yourself and go step by step. So far, it is still really requires the person to sit and do more work. It's not click and answer, but those steps, as if you understand the fundamentals from each step, you get to it. Once you have done your annotation, and you use other tools to do pathway analysis, network analysis, to try and make any biological sense of what you have measured. Yeah, thank you.
Okay, we have one last one here. It's not really a question. It's just someone saying, great work. I wish our lab collaborate with your lab. So maybe get in touch and you can collaborate.
No, we are very open for collaboration. And as I said really from the start, even some of the work that you saw, it's not just our lab, it's also with collaboration with people from other labs in Europe, US, and other parts of Africa also.
All right. I think we have no more questions. So I would like to thank very much your presentation. Great work. It was a very nice talk. And I would also like to thank all of you for staying to the end. So please connect with us and just register for the next month. Our next webinar will be in the middle of the month because it's before the Metabolomics conference. It's a warm-up with Monica Kala. The registration is already open, so just go to our LinkedIn page, find and register for the next one. And again, thanks, Fidele, and see you in Buenos Aires.
Thank you so much and see you soon.
Yeah.
Thank you very much, everyone. Bye-bye.
Bye-bye.
Thanks. Bye.